Friends, welcome to Biology Made Easy. Today in this video, we'll discuss Mendel and his experiments. Now in the history of science, July 22nd, 1822 is a red letter day. Why? Because it is this day the father of genetics, Gregor Johann Mendel, was born. So it is July 22 and 1822. And he was born in a very small village in Austria, and now that is known as Czech Republic. Though he was born to a very poor farmer's family, from the very childhood, he was very brilliant. During his school time, he read philosophy for several years, and at the age of 21, he joined as a monk in a monastery at Brunn. At the age of 25, that means in 1847, he was made priest of that monastery. But in 1851, he was sent to University of Vienna. To read botany and physics. Now, after completing the course in botany and physics, he returned back and joined as a teacher in the monastery at Brunn. Now, here, after joining as a teacher, where he worked for 14 years, the prefix Gregor, we are telling that Gregor Johann Mendel. Now this Gregor prefix was added only after he joined in the monastery as a teacher at Brunn. Now in this monastery, he worked as a teacher for 14 years. And while working as a teacher, he found that in the garden of that monastery, there are some pea plants where some contrasting characters are there. And these contrasting characters are not found in other plants. Like some plants are quite tall, some plants are quite dwarf. Some plants have round seeds, while some plants have wrinkled seeds. Some plants have axillary flowers, some have terminal flowers. In the same plant, both axillary and terminal are not there. So, as a teacher of physics and natural science, he was very much interested to know why all these things are happening in pea plant. That is why he started working in the pea plant. So he started working in pea plant, whose botanical name is Pisum sativum. Now in this plant, initially he took 34 pairs of characters, but found some differences. Then he reduced it to 22 pairs. Then finally he reduced to 7 pairs of characters. So ultimately Mendel worked on only 7 pairs of contrasting characters in the pea plant. Now one question comes to our mind. Why he selected the pea plant? It is because the pea plant has several advantages over other plants. So what are the reasons? That means we have to discuss reasons for choosing pea plant. What are the different reasons? Now one reason is, in pea plant, he got only pure varieties. That means there is no mixing of characters, and there are distinct contrasting characters. That means either the plants are tall or dwarf. These cells are either round or wrinkled. So completely distinct contrasting characters are there. Then the next point is flowers are bisexual. And another point is it is predominantly predominantly self-pollinated because it has bisexual flowers, so naturally it is self-pollinated. Now, another important point is, though it is predominantly self-pollinated, the cross-hybridization can be easily made. Cross-hybridization easily made. Then, the another important point is, there is no aftercare. That means after planting the young seedlings, you do not have to take much care. That is an important point. Next, the plants are annual plants. The plants are annual plants with only 2 to 3 months life cycle. That means in a single year, we can cultivate it 3 to 4 times. Then, large number of seeds are obtained from the plant. Large number of seeds are obtained, and another important point is, the F1 plants are fertile. This is a very important point, because usually the F1 hybrids become sterile, but here the F1 plants are fertile. So these are some of the points for which he chose the pea plant for his experimental purpose. Now, the next question comes to our mind. What are the 7 pairs of characters he chose for his experiment? So we have to answer that question. So the 7 pairs of characters which he chose, let us discuss here. Now the 7 pairs, now if you write here the serial number, then character, then the dominant, then recessive, and then the chromosome number in which the character is present. Now, number 1 character is stem height. Stem height. In stem height, the tall is a dominant character which is designated by capital T, and dwarf is a recessive character, small t, and it is present in chromosome number 4. Then the second one is flower color. Flower color may be violet, that is dominant character, or it may be white, then small v, and it is present in chromosome number 1. Then the third one is flower position. Flower position may be axial, it is dominant, or it may be terminal, small a, and it is also present in chromosome number 4. Then the fourth one is pod shape. The pod shape, it may be inflated, that means a capital I, or it may be constricted, small I, it is also present in chromosome 4. So here the pod is inflated or constricted. Inflated means it is just like this, it is inflated, but constricted means like this, it is constricted. This is the difference. Then the next one, then it is pod color. Pod color may be green, which is dominant, G, or it may be yellow, then it is small g, it is present in chromosome number 5. Then the next is seed shape. It may be round, capital R, because dominant, or it may be wrinkled, small r, and present in chromosome number 7. And the last is the seed color. The seed color it may be yellow, which is dominant, capital Y, or it may be green, small y, and it is present in chromosome number 1. So here this seed shape is round or wrinkled. This seed is like, if this like this is round, but if it is wrinkled, like this it is wrinkled. So from this table you have to remember which is dominant, which is recessive, and again the character is present in which chromosome, that is again very important. You see, first there is stem height. In the stem there will be flowers. In flower also there is a position and the color. Then the flower will be converted to fruit, which is known as pod. The pod has also shape and color. Then within the pod there is a seed. The seed has the shape and the color. So you can remember like this. And you can remember the number of chromosomes. There are 7 digits. Usually you remember 10 digits for anybody's phone number. So you can remember that it is 40, 41, 44 and 571. 41 lakhs, 44,000, 571. Or if you will add 3 digits, 000, that is the phone number of your friend, or you can tell it the phone number of Mendel. So very easily you can remember like this. And another important question is asked here. These 7 pairs of characters are present in how many chromosomes? Very simple, you see. They are present only in 4 chromosomes. 4 chromosomes. And what are the chromosome numbers? One is 1, 4, 5, 7. These are the 4 chromosomes where all the 7 pairs of characters are present. You see, in chromosome 4 there are 3 characters. So these are the important questions. So now these are the different types of characters he chose for his experiments. Now the next thing that we will discuss, why Mendel was very successful in his works. That point also we have to discuss. Reasons for his success, because from all these aspects usually questions are asked in examinations. So the next point we'll discuss is reasons for success. These are the things that we have to make. Now, first thing is, he took only pure breeding varieties, or you can tell pure lines. That is one of the important reasons. Took pure lines. So, since he selected 7 pairs of characters, that means the 14 varieties of pure lines were taken. That means suppose one contrasting pair is tall and dwarf. The tall is pure, the dwarf is pure. That means when the tall plant will be selfed, it will give rise to only tall. That means it is homologous tall. Similarly, for all the 14 varieties he found, they are true breeding. That means they are not giving rise to any other plant. That is number 1. The second point is there is a distinct contrasting characters were there. That means there is no mixing of characters. Only the characters were contrasting. That is another important point. Then during his experiment, he took only 1 or 2 characters at a time. He didn't take 3, 4 characters like that. He took only 1 character or 2 characters. Then the other thing is, the same experiment in different plants. This is also a very important point. That means suppose he made a cross between tall and dwarf plant and took 2 plants and found the results. Then in another set of experiment, tall dwarf, the same experiment. That means several plants, the same experiment is conducted and he found that the results are same in all cases. So that is also an important point. Then another thing is he made a reciprocal crosses. Now what is reciprocal cross? Now suppose we will take a tall plant and we will cross it with a dwarf plant. Suppose this is female and this is male, and the F1 hybrid is tall. But one may think, if we will take this plant as female and this plant as male, what will happen? Same. Because he conducted the reciprocal process. Suppose 50% of flowers of this plant will act as female flowers and this will act as male flowers. Then for the remaining 50%, the flowers of this will act as female and this as male. That means in the same plant, 50% flowers are female, 50% are male. That is a reciprocal cross. So he conducted the reciprocal process. Then next point is, he studied the inheritance for 2 to 3 generations. That means he studied F1, F2, F3 like that, and he found that the results are correct. That is one of the important points. Then the next point is the population size. Population size was large. That means in some of the experiments he grew thousands of plants. That means there is no doubt. If one will take only few plants, some hundreds, then there may be doubt. But he took sometimes some thousands of plants, large number of experimental materials. Then he put record of all experiments. All experiments he put record and applied statistical methods and mathematical analysis. That means he applied mathematics, that is algebra, and the theory of probability from statistics. And the most important point is, we can tell that Mendel was lucky. This is also a very important word for Mendel, because in science usually the luck word is not used, but this word is used for Mendel. Why? The 7 pairs of characters which he chose, they are located in only 4 chromosomes. So usually the characters he choose, they are present in either different chromosomes, or if in the same chromosome they are present far apart from each other, so that there is recombination, there is a separation, there is independent assortment. But he didn't take a pair of contrasting characters which are very close to each other. And what is that pair? That pair is the plant height and pod shape. You see, the plant height or stem height is in chromosome number 4 and the pod shape is also chromosome number 4. So these characters are present in chromosome number 4, but they are very close to each other. That is why always they do not behave independently. There is no independent assortment. And to his good fortune, he has never taken this pair of character for the dihybrid process. But you see, that same is that flower position is 4, while the stem height is in the 4, but they are far apart from each other. So for this reason, the word luck is used for him. And he was very much successful. Now the next point is, he was very successful in his experiments, but why he was not recognized at that time? That is also one of the important points. So reasons for non-recognition. So the next point that we'll discuss is reasons for non-recognition. Why he was not recognized, even if he was very much successful in his work. Now, the first thing is, after conducting experiments, because he conducted experiment for 7 years, that is 1856 to 1863, for 7 years. For 7 years he conducted his experiments. And in 1865, in 2 separate meetings, he read his formulations in the Natural History Society. In the Natural History Society, he put forth his formulations, and in 1866, that was published in the annual proceedings of the Natural History Society. But the other point is, this journal, it has no wide publication. It has no wide publication. That is why most of the scientists, they could not know that such work has been done. That is also one of the important things. Then the third point is, he failed in hawkweed. Now, hawkweed is another plant, and you know, Nageli is another scientist. Nageli suggested him that along with the pea plant also you work with hawkweed. But when Mendel conducted his experiment in hawkweed, he was not successful. So his confidence became down. That is also one of the reasons. Then the next point is, Mendel lacks aggressiveness in his personality. Now, you know, different types of persons are there. There are some people, they can contact so many people, but there are some people who are very shy, they cannot contact. And Mendel was like that. He could not contact most of the scientists, and he could not convince also most of the scientists. So that is also one of the important points. Then another important point is that factors are discrete, stable, on blending. This was not acceptable to the scientists at that time. They didn't accept. And another point is, no physical proof of factors. So the factors are actually transmitting from generation to generation. There is no physical proof. So scientists were not convinced. Then another point is, Mendel used mathematics and statistics to his experiments. This was ahead of time. That means the scientists who were contemporary to Mendel, they are not acquainted with the mathematics and statistics that will be applied to biology. So Mendel's work was ahead of time. The scientists could not believe that why the mathematics and statistics is applied to biology. So they didn't accept. And to all these things, another important reason is there. And the reason is Charles Darwin. During the same time, the 1856-1906, during the same time, Charles Darwin was working on the theory of evolution. But his personality was aggressive. He contacted most of the scientists throughout the world that how the evolution of human being has taken place. So all the scientists were busy at that time thinking how the evolution of human being was taking place, and nobody gave attention to Mendel's work. So unrecognized, he died in 1884 without getting any recognition. Then this Mendel's work was rediscovered in 1900. That means 16 years after his death. So then we have to discuss that how it was rediscovered. In 1900, three different scientists in different parts of the world, they were working independently and they came to the same conclusion when Mendel's formulations were rediscovered. So the next point is rediscovery of Mendel's work. So in 1900, that means 16 years after his death, it was Hugo de Vries of Holland, then Karl Correns from Germany, and Erich von Sarmack from Austria. These three scientists were working independently in the line of Mendel's work. And when they found that similar type of work has been done long before. So in this way, Mendel's work was rediscovered in 1900. And ultimately, in 1901, in 1901, Hugo de Vries published Mendel's works in the international journal Flora. Flora is one international journal. He published his work. Then, in the same year, Correns he converted Mendel's formulations to laws of inheritance. That means only after rediscovery, his formulations were known as laws of inheritance. And finally, Bateson and Ponnet, they observed that Mendel's laws, they are not only applicable to plants, they are also applicable to animals. Because Bateson and Ponnet, they have worked both in plants and animals. So his work is applicable both to plants and animals. Only after all these works, Mendel was given the title that is father of genetics. So this is how the Mendel has worked, his life, his working pattern, the characters, why he was successful, and even after that he was not recognized. So these are the very important things. Now let us discuss MCQs. So one MCQ can take in dihybrid cross. In dihybrid cross, which of the following pair of characters was never taken by Mendel? So one choice is plant height and pod color. Second is plant height and pod shape. The third is plant height and flower position. The next choice is pod shape and flower position. So which pair of character was not taken for his experiments? Now come to the second question. Who is not associated with rediscovery of Mendel's work? So one choice is Hugo de Vries, then B choice Bateson, third choice Correns, and fourth choice Sarmak. So I hope you have clearly understood the life, the working pattern, the characters, his success, and even after all these things, why he was not recognized. So thank you very much for watching the video. God bless you. Have a good day.